Okay, everyone, thank you for coming. I know we are getting to the end of this, so thank you that you managed to be around. I'm very proud to be chairing this session. It's one of the very important sessions that the IHS has put on the program. This is the IHS Grant section. I say we because I'm on the board of trustees of the International Headache Society, so we have a great interest in investing in new people who can do better research for the future of the headaches. So without any further ado, I would like to ask our first speaker to come to the stage. The first speaker is Carlene Moore from United States, Dr. Carlene Moore, who received the Child and Adolescent Research Grant from the IHS. And she will be presenting the New Delhi Persistent Headache as a biomarker study in children and adolescents.
Hi, good afternoon, everybody. Okay. I'll start with my disclosures, just one, and the overview. So the goal of this talk is to determine the clinical differences between new daily persistent headache and chronic migraine in children and adolescents. And the main objective of this study was to identify biomarkers that differentiate NDPH from chronic migraine and healthy controls. Then I'm going to go through my methodology. And the importance of this study was it's one of the very first biomarker study of NDPH biomarker study of NDPH and chronic migraine in pediatric populations. So what's the difference between chronic migraine and new daily persistent headache? It begins with the onset. Chronic migraine, it usually transforms from episodic migraine to chronic migraine, whereas new daily persistent headache, it has an abrupt onset, and it's continuous and unremitting from day one. Frequency for chronic migraine tends to have greater than 15 headache days per month, and it usually have to have had this for over 3 months and have to have had 8 or more migrainous features. With new daily persistent headache, it's daily, unremitting from the onset, and it persists greater than 3 months. Duration, greater, usually lasts 4 hours or more, and it's often variable. Whereas new daily persistent headache, it's always there, it's constant, and it's continuous, but there are exacerbations. Symptoms, headache, nausea, the common photophobia, phonophobia, osmophobia, whereas new daily headache might have some of these migrainous features, but they're not required to be identified as new daily persistent headache. History. Usually people who develop chronic migraine have had a history of some type of migraine, whereas new daily persistent headache, they have no history before the headache begins. And for chronic migraine, it's intermittent, and it usually improves with therapy. However, with new daily persistent headache, this can last years, and it's often refractory. Prevalence in youth, when we're looking at the youth population, chronic migraine accounts for 40 to 60% of headache cases in adolescents. So all these, when adolescents have migraine, 40 to 60% of them would end up with chronic migraine, whereas with new daily persistent headache, only 10 to 20% of them have new daily persistent headache. But in the adolescent phase, when you look at the prevalence of new daily persistent headache, it's actually more prevalent in adolescents than it is in adults. Treatment response, not very much out there. Chronic migraine, it often responds to most of the preventatives, and also the monoclonal CGRP antibodies and Botox. As I mentioned before, it's frequently refractory. New daily persistent headache, it's frequently refractory, and there is occasional benefit from the Botox and the monoclonal antibodies. So here is an outline of our study that we did with the money that we received from the International Headache Society. The study lasted 1 year, and we had 13 healthy control participants, 12 chronic migraine, and 13 NDPH patients. And as you could see, the age range were for healthy controls, our median age was 13, and 15 for chronic migraine and NDPH. And we had, as you could see, our data was pretty much imbalanced. We had 6 male healthy controls, 7 females. We did not have any, and I'll discuss this a little later, we didn't have any male chronic migraine, and we had 2 females and 11, 2 males and 11 females in our NDPH group. Most of these patients were recruited in the Duke Pediatric Clinic and our headache clinic, or via the electronic health screening, or especially for the controls, we went out into the community and networks just to find healthy controls. Actually, those were the hardest ones to get. Baseline assessments, we did blood draws, and then we did a number of different scoring, like the PED-MIDAS, the PHQ-9, and the SCARED screening. And then we did our biomarker analysis, where we looked for a number of cytokine. We used the MS Mesoscale Diagnostic cytokine panel and a BioLegend Plex, where we used 13 cytokines. And then we did a number of ELISAs for CGRP, PACAP, VIP, the whole thing. And then we did our analysis. And here's a list of all of the biomarkers that we actually assayed for. So I'm going to go a little bit into what's known about CGRP levels in NDPH, what we know and what we don't know. So everybody knows what CGRP is, and it has been shown to be elevated in migraine and other primary headache disorders. And blocking CGRP signaling have been shown to alleviate pain in many patients. However, there is no published CGRP levels in NDPH, and so that's one of the reasons why we looked for CGRP in New Delhi persistent headache. And we saw one study when I was looking to make this presentation. There was one study that was proposed to assay CGRP and NGF levels in the New Delhi persistent headache, but I don't think this study went to completion because of COVID. I think it was stopped. And when we think about the clinical relevance of New Delhi persistent headache and CGRP, some studies, there was one study that came out recently by Chima et al. That was just published in the Journal of Headache and Pain, where they said, they showed that the monoclonal antibodies for CGRP, there was moderate benefit. And then there was also another case report that came out this year that showed that this one patient had complete remission with erunamab in the New Delhi persistent headache. So what did we find in our study with CGRP? Here is our data. We found an elevation of CGRP in our New Delhi persistent headache patients. And this was higher in our New Delhi persistent headache than the chronic migraine. As you could see, we saw an increase in CGRP in the chronic migraine, but that was not statistically significant because our study was underpowered, I think. But I think once we get more numbers, we probably might get to decent power. But however, we saw an elevated amount of CGRP in these New Delhi persistent headache patients. And because, as I mentioned, our study was so imbalanced by data, I decided to separate the data by male and female. And in either case, CGRP was still elevated in just even in the females, and when we separated with just the 2 males we had, CGRP was still elevated in these New Delhi persistent headache patients. So we also found, and I'm only reporting the ones that we saw significant changes in. We saw significant changes in IL-6, IL-8, and TNF-alpha, although these changes were between chronic migraine and New Delhi persistent headache patients. As you could see, especially for IL-8, we had like a wide spread in our controls, but we didn't see significant elevation between control and any of these patient population, but we were able to see differences in chronic migraine and new daily persistent headache. So what's out there? What information is out there about these cytokines, chemokines in new daily persistent headache? One study found higher levels of IL-6. This was not a study, actually. This was a meta-analysis. And they went through all the literature and looked for the changes in cytokines in daily persistent headache. And they found that in headache in general, IL-6, IL-8, and TNF was found to be elevated in migraine and in migraine only compared to controls, and they didn't even find anything about new daily persistent headache because they just did not have enough literature. There was another recent biomarker study looking at these same cytokines in new daily persistent headache, and they've showed that elevated IL-6 in NDPH and chronic migraine, but not in healthy controls, and then they did not see any difference in IL-6 and TNF-alpha. So these findings, including mine, suggest persistent neuroinflammation in new daily persistent headache pathophysiology, and these cytokines could potentially serve as biomarkers for new daily persistent headache, especially when you're comparing with chronic migraine. The next one, and the surprising thing that we found in this study, we saw an elevation of this STREM2 in our chronic migraine patients. And if you look at the numbers, it's very elevated in the chronic migraine patients compared to healthy control and new daily persistent headache. And when we separated by females, it was still statistically significant in the chronic migraine patients. However, well, of course, when we look at the males, we did not have any chronic migraine in the, we did not have any chronic migraine males. So what is STREM2 and why I think it's important? It is soluble form of triggering receptor expressed in myeloid cells, TREM2, and it is generated by proteolytic cleavage from microglia, and it has been shown to regulate microglial survival, proliferation, and inflammatory signaling. And what it's actually known for is most neurodegenerative diseases. It's actually been considered as a biomarker in dementia and Alzheimer's disease, which is, I think this is going to be interesting. So what has been known about TREM2 and migraine? We know that microglial activation and inflammatory pathways are implicated in migraine pathophysiology. In this one study, using mice, TREM2 levels were found to be elevated in the mouse model of migraine and was attenuated with metformin, and this attenuation actually blocks central sensitization via this TREM2-SIG pathway. And here is some of their information when they, some of their biochemistry, protein biochemistry, when they looked at TREM2 levels over time with treatment of NTG, which is a mouse migraine model, and they saw an increase of TREM2 in these animal models. So this data, along with my data, suggests that TREM2 is an emerging link between microbiology, neuroinflammation, and migraine, and also may serve as a potential biomarker of these processes. So what my study showed, our study is the first to demonstrate elevated CGRP in pediatric new daily persistent headache. Pediatric patients with new daily persistent headache had elevated TNF-alpha, ILH, and IL-6 compared to chronic migraine patients. Soluble TREM2 was significantly elevated in chronic migraine compared to healthy control and new daily persistent headache paper. So, and one thing that this data clearly shows is that NDPH is simply not a continuous migraine, but a headache with distinct biomarker profile. But further studies with larger cohorts and standardized methodologies like age and sex match and everything else are needed to validate the diagnostic utility of these markers. I want to thank the International Headache Society for the funding. I also wanted to thank Carol Taylor, who's been invaluable, helpful to us. I also wanted to thank Dr. Klaus Werner. He's the pediatric neurologist who recruited and diagnosed all the patients. And here is my team, who's been instrumental in doing with all the biomarker analysis, blood, working with the blood and everything. So I just want to thank him and thank you. That's it.
Congratulations, first of all. Very nice project.
Thank you.
Do we have any questions from the audience? No. Can I ask? We do. Yes, please. Finally, someone will ask us a question. Oh, there is a microphone over there.
Is it of common knowledge of neuropediatrics doctors these differences between NPHD and migraine?
Dr. Werner is back there, but he swears that most neurologists who treat patients should know the difference, because there is one qualifying features of new daily persistent headache is the patients always remember the day, the time when the headache started. And so if they're asking the correct questions, they should be able to differentiate between new daily persistent headache and chronic migraine.
Any other questions? Can I ask you a question?
Sure.
So with biomarkers, especially with the assays looking at the peptides, not to mention the interleukins, usually you need big numbers to actually see a trend. So how did you manage? What's your trick? You use a platform that we've been using in adults, and we need to recreate 70 patients to see a trend. So with such a small number of pediatric population, you've seen
we were just as surprised as you guys. And what we did, though, we were very diligent with how we collected the samples.
When did you collect the samples?
I mean, once he saw the patients, he would call me and somebody from my lab, and we would be there as soon as the blood comes out the patients. Within an hour,
and
that
so
blood was...
That wasn't a specific time of the day?
No, it wasn't so specific, but within an hour, that blood was in -80, frozen back. So we were, because we know these biomarkers as degrade so quickly, we were very diligent in, keeping them cold and getting them in -80 really fast.
I still insist that there must be something else, because that's what we do. We always keep it very fast. You think, my question is, have you tried to compare the levels you recorded there in the pediatric population to adults, I guess? Because maybe the answer is that children have higher levels.
Especially for CGRP, we did 2 kits. We did 2 separate ELISAs, because when I first saw it, I was like, okay, I can't go and report this. This is, let me just try another ELISA from another company, and the data was exactly the same.
Okay.
And also with the mesoscale and the BioLegend, a lot of those cytokines overlap, like IL-6 and the TNF-alpha, and we saw very similar results. So I wanted to verify. I was like, I can't just go over there with just one time doing it. And so I kind of repeated them. So yes.
Thank you very much. Very interesting data. Thank you.
Thank you.
Oh, sorry, we had one more. I will tell you, because we have time, because some of the speakers are not here. Yes, please, we can take that one.
And I can share which kits we used.
Yes, please.
To the patients, found etiology viral to the patients, or you research etiologies for headache?
What's the reason they develop NADH, you mean?
Yes, the etiology to headache. CU pinoviral or LE.
I spoke to Klaus about that, because he's the one who diagnosed all the patients, and he said there was nothing that could explain why the literature says sometimes it's, the literature says sometimes it's from febrile illness or some type of sickness, maybe even COVID but from him, he said there was nothing for most of these kids. They just developed this and it just wouldn't go away.
Okay, thank you.
Okay, thank you. Thank you so much. Lovely presentation. All right, we can move on with the next presentation from Annemijn Oosterlee from the Netherlands. Annemijn won an IHS Junior Grant for the home-based EEG for early detection of brain state alterations before the migraine attack. Would that be a biomarker as well? So maybe we can hear that. Thank you.
Thank you. Let's see. All right, so my name is Annemijn Oosterlee, and I'm a PhD candidate from the Leiden University Medical Center in the Netherlands. And indeed, I'm going to tell you something about home-based EEG and how we can use that to predict migraine attacks. Before I do that, these are my disclosures. And before I tell you about all the projects we did, I'd like to tell you about the aim, the aim that we have, which is predicting migraine attacks. And how we want to do that is by measuring pre-ictal cortical excitability changes via EEG response to visual stimuli. As you can see here on the pictures, the visual stimuli are emitted through LED goggles that we put on the patient. One important aspect of this during our project is that we wanted to transform the recordings from the hospital setting to the home environment of the patient. And in the future, we hope if we're able to predict those attacks, so patients know when an attack is coming, that they can use situational prevention by taking G-pens, for example, instead of having to take daily preventive medication. So what are we looking for? We are looking for EEG biomarkers in the pre-ictal phase of a migraine attack. We largely know what happens during the ictal phase of an attack, so the aura and headache phase. But what remains more unclear is what happens during the pre-ictal or prodromal phase. And we hope to find early warning signals that we can use as an alarm tool that an attack is coming. Before I tell you about the 3 projects that we did to achieve this goal, one small note about the EEG systems that we used. Because on the one hand you have the gel-based systems, and these were the first systems we used. The signal quality is the highest in this system. They are the gold standard for EEG systems. However, the patient friendliness, the usability to be able to perform your own measurements is quite low because patients couldn't quite reach the back of their head and put the gel in the right way in the electrodes. On the other hand of the spectrum, you have the dry electrodes. They are highly patient friendly as you can just put the headband on. However, the signal quality is way worse, so we couldn't really use that. And we think we have found that we've struck the right balance between signal quality and usability by making use of a water-based system. And for this, patients can just wet some sponges, put it in the cap, and then put the cap on. So it's highly patient-friendly. They can do it themselves at home. And we found that the signal quality is comparable to that of the gold standard, which is the gel-based systems. And this is the long road we've taken or are still taking towards home-based EEG. We started with a hospital-based study that was cross-sectional. We then converted that to longitudinal measurements, still in the hospital. We then made the switch to home-based measurements. These were still longitudinal and supervised and performed by the researcher. And in the end, we hope to have home-based measurements that patients can independently perform themselves. So on to the first study. We made use of a chirp paradigm, and that means that the stimulation frequencies that we put in were between 10 and 40 hertz. So the visual stimuli, which were light flashes emitted through the LED goggles that patients put on, that stimulation frequency was between 10 and 40 hertz. And we then looked at the oscillation of the brain, so the reaction of the brain towards these stimulation frequencies, and we measured that in EEG response power, which is measured in mean decibel change from baseline. I hope this works. This is what the patients see. So this is the chirp paradigm. And they have to close their eyes during the measurements because it's really bright when it's like here. All right, so the first study was completed by one of my predecessors, Thijs Perenboom. He compared healthy controls with migraine with aura and migraine without aura patients to see if he could find EEG response power differences when visual stimuli were performed. While he did not find a difference between the healthy controls and the migraine patients, he did find something else. So if we look at the harmonic frequencies, if you look on the x-axis, you can see the stimulation frequencies that I talked about, so between the 10 and 40 hertz, what we put in, and on the y-axis, you can see what the brain puts out, so the reaction, the EEG response power. And the black line, that is actually the pre-ictal phase, while the blue line is the interictal phase. So you can see the difference between those two, and that there was a significantly higher EEG response power in the pre-ictal phase. And this is what really put us on the path to finding if that could be a useful biomarker to identify upcoming attacks. So our next step was converting the study to a longitudinal design. This is what my predecessor Thomas van der Hoek did, and this is what he received the IHS Junior Research Grant for and what it was largely spent on. He included female migraine patients who had menstruation-related migraine, so he roughly knew or could estimate when the next attack was going to happen, and he started recording 6 days before that and could record patients daily for a maximum of 6 days. The measurements themselves took about 20 minutes per day. And before I tell you the results of this study, I'd like to tell you why during this protocol we switched from hospital-based to home-based measurements. Because we found that the hospital-based measurements were quite intensive for the patients, and it wasn't really representative of their daily surroundings, which of course their home is. And we also added our validated and time-stamped e-diary, which I have a picture of here, because we now had a measurement for migraine susceptibility, which is the EEG of course, but we also needed to be able to identify pre-ictal from interictal days. So we knew when our measurement was truly pre-ictal and when it was interictal. Because if we want to find a difference in the pre-ictal phase, then we need something to compare it to, and that is, of course, an interictal measurement, or a normal day for the patient. So these measurements that we then started performing at home, the protocol was still the same, so we could record for a maximum of 6 days. This is what I did myself with my colleague from the Technical University Delft in the Netherlands, Jamie Duivelshof. We included episodic migraine patients who had at least 1 to 2 attacks per month, and we started measuring them for a maximum of 6 days, which meant that I visited the home of the patient every day. Which, as you can imagine, is quite labor-intensive. Here you can see me leaving the hospital with the suitcase, getting in my car and performing the measurements at the patient's homes. This was quite a lot of work, so we could not keep this up forever. And these are the first results from those hospital-based and home-based longitudinal recordings I'd like to share with you. We screened over 80 patients during a period of little more than 2 years. Of those, we were able to include 30 participants, and we had over 150 measurements. However, most of those measurements were mostly interictal, and we needed at least 1 pre-ictal measurement per person. And adding on to that, we had some technical issues with the pre-processing of the EEG data, which we have now resolved, so we'll be able to use that data in the future, but for now we couldn't. This led us to being able to include 17 participants who had at least 1 interictal and 1 pre-ictal measurement. Here you can see their demographics. They were overwhelmingly female, had a mean age of 52, mostly had migraine without aura, and most of them did not use any prophylactics. If they did, it was either candesartan, iranimab, or topiramate. And these are the results. I'd like to focus on the big picture here. You can see the stimulation frequencies again on the x-axis, so what we put in, and on the y-axis the EEG response power, which is measured in mean decibel change from baseline. And the red line here represents the interictal phase, while the blue line represents the pre-ictal phase. And as you maybe have already seen, we were not able to replicate the results of the cross-sectional study, where they did find the difference between the phases. I've highlighted the band of interest here, which is between 22 and 32 hertz, where they did find the difference in the cross-sectional study. But here we looked at the different frequencies, and we were not able to replicate those results. And we think we know why this happened, and that is that we had a power problem. Our sample size was simply too small, and there are 2 main aspects to that. First of all, we had too few people. People found it quite challenging to come to the hospital every day and were not as motivated to do so by the end. And we also had a logistical problem where I had to, or another researcher had to go to the patients' homes, and this sometimes coincided with their own job or other appointments that they had, so that made it quite the logistical challenge. Another important aspect is that we had too few pre-ictal measurements. As I mentioned before, we had a lot of people that had only interictal measurements, and we think for the people that came to the hospital, this had to do with the fact that the added stress of having to come to the hospital every day might have postponed the attack, and after 6 days we had to stop measuring because we could only measure for 6 days as was in the protocol. And again, the logistical problem made it that we could not measure any longer, so had a slightly or largely lower chance of finding pre-ictal measurements. Luckily, we have found a solution, we think, to this problem, and that project is called Migraine at Home. This is the project that I'm currently working on, and for this we have developed our own EEG setup, which patients can take home with them and perform the measurements independently, so themselves. And we added the water cap that I discussed before to this, as well as a tablet, which guides patients through the measurements and helps them perform it, and at the end sends the EEG data directly to us. We strive to include 200 patients in this study, who will all receive an instruction at our hospital, after which they will receive the setup in a smaller suitcase than before to their home, and there they can measure themselves for at least a month. And we think this will tackle the power problem, as we will include 1, way more people, and 2, will measure for a much longer time, which will help us hopefully to catch more pre-ictal measurements. And the fact that people can perform their own measurements in the comfort of their own home also tackles the stress and logistical challenges that we had before. So we've already included 20 participants that have successfully measured themselves in the comfort of their own home, and we've received their data. So we aim to include 180 more to eventually be able to predict migraine attacks by identifying excitability changes in the pre-ictal phase. For now, I'd like to thank the IHS, and I'd like to thank my team, and thank you all for listening.
Thank you for the lovely presentation, and congratulations. A very ambitious project.
Thank you.
Do we have any questions from the audience? Is there any time of the day that you ask the patients to do the recording?
Yeah, we ask the patients to do it at the same time every day.
Does it matter what time they do it?
No, it doesn't matter to us what time it is. If it's at the same time every day, then it's fine by us.
Okay.
And we can also see in the measurements exactly what the date and the time and everything was, so it's time-stamped as well.
All right. The visual stimulation you showed will obviously activate the visual cortex. Do you think there will be any difference there, and does it matter what time of the day that will be?
Yeah, I think the largest chance that we have of finding a difference is in the visual cortex. As we all know that migraine patients often have photophobia, and of course the visual cortex is implicated in the aura phase as well. So that's why we chose to stimulate the visual cortex. And I think most patients measure themselves in the morning before their job. So that's what we advise them to do as well. But for some, that's just not feasible. And then we say it's okay if you measure yourselves at night.
Out of curiosity, of the recordings you did at the hospital compared to those that were done at home, was there any difference? Obviously, small number, maybe the signal is too small to be detected, but I wonder whether the hospital environment indeed will have a different effect.
Yeah, the hospital environment had a different effect in the way that the signal quality was different, because we had, I don't know if it's the right pronunciation, a cage of Faraday, so you had no outside stimulation. So the 50 hertz, the electrical current that we all have in our own homes.
So how do you go at home without a Faraday cage then?
We can filter that out.
Have you gifted them a Faraday cage?
No,
Wow,
we filter everything out at home. Yeah, because we can't ask of patients to lock themselves in a Faraday cage at home.
But I guess you advise them to stay away from other devices.
Yes, definitely.
Yeah. Wow, amazing. I'm really looking forward to see the outcome of this. Congratulations. Very good work. Okay, for the next speech, unfortunately, the speaker couldn't be
Hello,
here
everyone.
with us. Hello, can you hear us? Luigi, can you hear us? So we have a virtual presentation by Luigi Iannone in Italy, who won one of the Junior Research Grants. And unfortunately, he couldn't be here in present, but he kindly agreed to provide us with a virtual presentation. So Luigi, if you can hear us, thank you for being here, and please go ahead.
Hello, everyone, and thank you so much for the opportunity to present our data in the Congress about the Schwann cell pathway regulation by triptans and ditans. The idea of this project and the Junior Grant start with this paper from our group in 2022, when we demonstrated that the Schwann cell can have a role in mediating mechanical allodynia evoked by CGRP injection or capsaicin injection. And as you see in this illustration, there is very complex crosstalk between C fiber and delta fiber mediated by Schwann cell through mainly the CGRP receptor and several trip receptor like the vanilloid 1 and A1 receptor. The Schwann cell we are talking about are just one type that we have and are the REMAC Schwann cells, so the non-myelinizating Schwann cells that constitute the REMAC bundle in the nerve. So the main hypothesis of our study is that triptan and ditans mediate, at least in part, allodynia in mice by reducing the increase of cyclic AMP evoked by CGRP or capsaicin in Schwann cell that are surrounding the trigeminal nerve fibers. To do so, we planned several aims with both experiment in vitro and in vivo. In vitro, we plan to better identify the expression of serotonin receptor in mouse and human Schwann cell using RNA scope and immunofluorescence, and verify if this receptor using their specific agonist can attenuate the CMP intracellular levels both in human and mice Schwann cells using cyclic MPSAs and calcium imaging. To confirm this data, we planned, as I said, some experiments in vivo using mainly, but not just, but mainly the periorbital mechanical allodynia using the von Frey needles and the top and down method after injection of CGRP or capsaicin using both female and male mice. We used this method to explore if this receptor agonist can attenuate the PMA in mice and if this PMA is cause of allodynia but not acute spontaneous pain. I would like to underline that when we started the study, it was very challenging to choose the right molecules and the right doses because we are aware in clinic that triptans are very different among them. But when we need to choose a specific dose and a specific molecule for a different receptor, it will be very challenging. So we used the different doses and different molecules to better understand our pathway. So talking about results in vitro, we first confirmed the presence of serotonin receptor 1B, 1D, and 1F in both human and mouse Schwann cells at different concentration, as you see expressed as relative expression of mRNA, but we show that are present in both human and mouse Schwann cell using RNA scope. We confirmed this data also using immunofluorescence, as you see here, again, in both human and mouse Schwann cell. In green, you can see the localization of the receptors 1B, 1D, and 1F, then the localization of the marker of the Schwann cell S100, and then the colocalization in the first in the first panel. So we confirm the expression with different techniques of serotonin receptor in Schwann cells. Then, moving to another objective in vitro, we also demonstrate that intracellular cIMP is reduced by the pretreatment with sumatriptan, but also with other triptans and lasmiditan. You see here the data about sumatriptan, and you see in red the expression of total CNP with CGRP, and then in green and blue the co-administration of CGRP and sumatriptan to different dosage with almost complete attenuation of total cyclic IMP with sumatriptan. Moving forward with in vivo results, where when we try to confirm this results, we use two different settings. First, we use the pretreatment with sumatriptan and other treatments and lasmiditan, using the von Frey technique to assess periorbital allodynia. When evoked by CGRP, you can see here the pretreatment with sumatriptan in both male and female attenuate almost completely the response with threshold of the mechanical allodynia that's similar to vehicle when pretreated with sumatriptan. The same pathway is reported in both male and females with just some slight differences. The same was confirmed using capsaicin to evoke mechanical allodynia. That, you know, capsaicin is an agonist or TRPV1. And as you see here, we have almost the same results with a complete attenuation of PMA with the pretreatment with sumatriptan. Moving forward, we also use post-treatment sumatriptan after the injection of CGRP or capsaicin, most mimicking better what happened in human. And as you see here, by CGRP, we have almost the same results. Then you can see the threshold that go down with the injection of CGRP, and then after treatment with sumatriptan, it comes back at the vehicle level. It's applied for both male and female. The same is confirmed once again with capsaicin, as you see the post-treatment with sumatriptan resolve the periorbital mechanical allodynia. Then we use several different triptans at several different dosages, including also lasmiditan at different dosage. Here we show the most significant one with sumatriptan once again, but with 3 different dosages. As you see, we have a very clear dose effect. The same applies for homotriptan in the center, and then lasmiditan on the right, when we use lots of different dosages, and just 2 dosages, the 1 micromole and the 100 nanomole dosages are statistically significant compared to all the CGRP injection, while the other dosages are not significantly different from CGRP, although there is a little attenuation of mechanical allodynia. So we repeated this data with also other triptans, other dosages, but for a matter of time, I don't show that. And what we can say is that we confirm the expression of serotonin receptor in mouse and human Schwann cell using different techniques. We show the ability of this receptor agonist to attenuate the increase of cyclic MP in vitro. And then we showed the same effect also in vivo using the periorbital mechanical allodynia model in mice. What we have done other data that go beyond the easy IHC grant, and some of that are still ongoing. Confirming the role of Schwann cell using selective selection, confirming the role of TRPA1 Schwann cell also in this pathway, and other tests including extending the sample size, male versus female, adding other behavioral tests more than the only allodemia. So I would like to thank so much the great group in Florence, in particular Professor Pierangelo Geppetti. Thank you for International Headache Society for the grant and for my National Society of Headache that sustain us all year. And unluckily, I'm not here to answer the question, but feel free to contact me by mail or Twitter, or see you in person on the IHC Congress.
I would like to thank the organizing committee for the opportunity to present the results of this project, which was partly supported by the International Headache Society through the Junior Research Grant in 2022. I will talk to you about the endocannabinoid system and TRP channels as potential therapeutic targets for sexual dimorphism in migraine. Here my disclosure of interest. As an introduction, migraine is a neurovascular disorder which is more prevalent in women than in men, who also present a higher risk of cardiovascular events. Among others, current treatment for migraine include targeting a CGRP pathway, which involve the use of triptans and monoclonal antibodies. Nevertheless, there is still a lone population that they don't respond to the current treatments. Moreover, there are also some concerns about the cardiovascular safety of blocking the CGRP pathway. In this respect, it has been suggested that TRP channels and endocannabinoids might represent new therapeutic options for the treatment of migraine. The endocannabinoid system plays an important role in pathophysiological functions, including the regulation of cardiovascular system and in inflammatory and chronic pain, including migraine. Endocannabinoids can activate canonical receptors, such as CB1 and CB2 receptors, and non-canonical receptors, including the GPR55 and the endothelial cannabinoid receptor, or CBE. In addition, it has been suggested that anandamide can also activate TRPV1 channels. Cannabinoid receptors are located in endothelial cells and in smooth muscle cells, where they modulate the vascular tone through different mechanisms. These receptors are also located in perivascular sensory nerve, where they regulate the release of CGRP to induce relaxation responses. What about the TRPV1 channels? In human in vivo models, topical application of capsaicin, which is a TRPV1 activator, induce the release of CGRP from sensory nerves to induce increases in dermal blood flow. These vascular responses are mediated through activation of CGRP, as it has been demonstrated that blockers for the CGRP receptor modulate these responses. In contrast, in human isolated blood vessels, including human coronary arteries or human middle meningeal arteries, the relaxation responses induced by capsaicin are not inhibited in the presence of capsaicin or olzigipam, which they are blocker for TRPV1 channels or CGRP receptors, respectively. These responses were similar in blood vessels from men and female. Then the question is, do cannabinoids modulate TRPV1-induced vasoactive effects? In this study, we use human coronary arteries. Distal segments of the human coronary arteries were mounted in multipany setups to assess the vascular reactivity in the presence of absence of different compounds. It's important to mention that although they are different, anatomically different from cerebral blood vessels or vessels that they are related to the trigeminal system, human coronary arteries share some characteristics, including that they express TRPV1 receptor channels and they respond to CGRP, which is a potent vasodilator involved in migraine pathophysiology. These characteristics suggest that human coronary arteries are a good model to study migraine-related mechanisms and also to evaluate potential cardiovascular safety of different drugs. In human coronary arteries from both male and females, capsaicin induced relaxation responses. These relaxation responses were inhibited in the presence of the antagonist for CB1 receptors, AM6545, but not in the presence of the antagonist for CB2 receptors or TRPV1 channels. When evaluating the responses by the combination of the antagonist for CB1 receptors and plus capsaicin, the responses were not further, there was not further effect. We also evaluate the effect of antagonists for the non-canonical receptors in the responses to capsaicin. The relaxation responses to capsaicin were inhibited only in the presence of the antagonist for the endothelial CB receptors, O1918, but not in the presence of the antagonist for the GPR55 receptors. These results suggest that capsaicin-induced vasodilatation in both a TRPV1 independent mechanisms, and it is mediated via the activation of CB1 receptors and the endothelial CB receptors. Continue with the analysis of the interaction between the endocannabinoid and the vanilloid system. We also evaluate the effects, the vascular effects of anandamide, which is a non-selective agonist for the CB receptors. Anandamide induced relaxation responses in both arteries for males and females. These responses were inhibited in the presence of the antagonist for CB1 receptors and in the presence of capsaicin. Similar results were observed when we investigate the vascular responses to ACA, which is a selective agonist for CB1 receptors. These results suggest that CB1-mediated vasodilatation might involve the activation of TRPV1 channels. To further explore this interaction between TRPV1 channels and the endocannabinoid system, we incubate segment arteries with capsaicin to induce desensitization of the TRPV1 channels. This incubation was during 1 hour, and then the relaxation responses to anandamide and also to ACA were evaluated. The relaxation responses to anandamide were inhibited after the incubation with capsaicin only in arteries from females, but not in arteries of males. Similar results were observed when investigating the relaxation responses to ACA. These results suggest that the vasodilatory effects of anandamide and ACA are partly mediated through activation of TRPV1 channels in females, but not in males. In this project, we also investigate the role of CB receptors in the modulation of vascular tone. For this part of the project, we use both human coronary arteries and human middle meningeal arteries, which they are a proxy for the trigeminal system. In human coronary arteries from both male and females, the relaxation responses to anandamide were inhibited in the presence of the antagonist for CB1 receptors, in the presence of capsaicin, and in the presence of olzigipine, which is an antagonist for the CGRP receptors. These responses to anandamide were also inhibited in the presence of the antagonist for the non-canonical receptors. Similar results in human coronary arteries were observed when analyzing the relaxation responses to ACA. In human middle meningeal arteries, we observed also similar results. The relaxation responses to anandamide were inhibited in the presence of the antagonist for CB1 receptors, in the presence of olzigipine, and in the presence of capsaicin. To prove that CB2 receptors are not involved in the relaxation responses to anandamide, we use a selective agonist for CB2 receptors. These agonists also induce relaxation responses, but these responses were not inhibited in the presence of the selective antagonist for CB2 receptors, in the presence of capsaicin, or in the presence of olzigipine. These results suggest that CB1 receptors mediate vasodilatation to anandamide and ACA, which might involve activation of TRPV1 channels and CGRP receptors, with no contribution from the CB2 receptors. The responses to anandamide and ACA were also compared between males and females. As you can see in this figure, anandamide and ACA induce and enhance relaxation responses in females when compared to those induced in males. In both human coronary arteries from males and females, these responses to anandamide and ACA were inhibited by the antagonist to the CB1 receptors. We also evaluate the expression of the protein for both CB1 and CB2 receptors. These experiments were developed by the technician Usha Muster-Babu. Both CB1 receptors and CB2 are expressed in human coronary arteries from both males and females. As you can see, CB1 receptors exhibit a higher expression in human coronary arteries from females when compared to males, and no sex differences were observed in the CB2 expression. These results suggest that CB1 receptor expression is higher in arteries from females than in males, potentially underlying the sex-dependent differences in vasodilatation induced by anandamide or ACA. Finally, capsaicin-induced vasodilatation is mediated via activation of CB1 and CBE receptors independently of TRPV1 channels or CB2 signaling. This is considering that capsaicin, or the antagonist, the selective antagonist for the CB2 receptor, didn't inhibit the relaxation responses to capsaicin in human coronary arteries. CB1 activation mediates vasodilatation in response to anandamide and ACA, potentially involving TRPV1 channels and CGRP receptors, with no contribution from the CB2 receptors. Endocannabinoids induce sex-dependent vasodilatation in human coronary arteries, considering that both anandamide and ACA induce enhanced relaxation responses in human coronary arteries from females than in males. This might be probably because of higher expression of the CB1 receptors. A dual interaction exists between the endocannabinoid and the vanilloid system in the modulation of the vascular tone, which might involve sex-dependent mechanisms. This is considering that after TRPV1 channel desensitization, the relaxation responses to anandamide and ACA were inhibited in human coronary arteries from females, but not in males. Probably there is an important role of sex hormones in these responses. Finally, targeting both CB1 receptors and TRPV1 channels could represent a promising therapeutic strategy for neurovascular disorders. I would like to thank all my colleagues involved in this project, especially to Professor Antoinette Massen-Vandenbrink for all her support. To the Secretaría de Educación, Ciencia, Tecnología e Innovación de la Ciudad de México for supporting this project, and the International Headache Society for the Junior Research Grant in 2022. I will also thank to Carol Taylor for all her support during this project. If you have any question, do not hesitate to contact me. Thank you very much for your attention.